So our webinar is on thoroughbred pregnancy and foal health. We have a great panel, as I mentioned. We have Kris Hughes from Sturt University, who will be presenting on the effects of fecal microbiota transplant therapy on infectious diarrhea in foals. Hello, Kris. We have Dr. Joan Carrick, who will be presenting some key findings identified in the pathology and epidemiology of equine pregnancy loss research project. And finally, we will have James Murray of Thoroughbred Breeders Australia on phase 2 of the post-mortem project. Now, a bit of housekeeping. As normal, we'll have everyone jump on, talk through their presentation, and then we'll ask any questions you have at the end. And if we can't get to your question, we will get back to you after the webinar. This webinar will be recorded, so you can watch it back later or send it to some of your friends that you might have. So I'm going to hand over tonight to Annalise to kick things off.
Thanks, ladies and gentlemen, for joining us tonight. I'm just going to do a brief run through about the Thoroughbred Horses program to give you a bit of background before we get into the presentations. So the Thoroughbred Horses program is one of 13 levied industries that are managed by AgriFutures Australia. The Thoroughbred Horses program invests levy funds on behalf of thoroughbred breeders. The levy consists of $10 from every mare returned paid by the mare owner and $10 from every mare served paid by the stallion owner. The levies are then matched by the Commonwealth Government at 50 cents for every dollar spent on research and development. Currently, the program has 24 ongoing projects, and some of our recent key achievements are a management of heat stress in stallions. We have a couple of really good documents on our website around that, and I'll put a link to our website in the chat. And then we also have done a quicker test for chlamydia and herpes. We have also invested in using advanced diagnostic imaging for thoroughbred horses in racing, and we also do a fair bit of industry support. We fund fast track positions, which is one of the Thoroughbred Breeders Australia initiatives. We have got a Vet Horizons scholar who is a vet student at the University of Queensland, and we also supported the Thoroughbred Welfare Initiative, which we discussed in our last webinar. So I think one of the big things that we're doing in the Thoroughbred Horses program at the moment is that we are planning our investments for the next 5 years. So we are putting together the Thoroughbred Horses program strategic plan for the next 5 years, which will guide our research and development for that 5 years. We have finished drafting it, but the most important part is yet to come, and that's the public consultation part. I really would appreciate if everybody could get online and join our newsletter, because the the public consultation for the 5-year plan will go out through our newsletter. So what we will do is we will get public consultation, we will get any comments back from anyone, and then we'll finish that off and we will release the next 5-year plan, hopefully in September. So as Charmaine noted, that's a very brief overview of the Thoroughbred Horses Levy program. We've got Kris talking next about transplant therapy. We've got Dr. Joan Carrick talking about pathology and epidemiology of pregnancy loss. And then we've got James from Thoroughbred Breeders Australia talking about the next phase of the post-mortem program. So I will now hand over to Kris. So thanks, Kris.
This is just a brief overview of a project that we've been running for the last couple of years through Sturt University, and this is looking at diarrhea in foals and looking at new strategies in which we may be able to manage this condition and hopefully get better outcomes and faster outcomes in terms of resolution of diarrhea. So this is the official title of the project, and what this really means is using fecal material, manure from a healthy horse, and transplanting that into an animal with infectious diarrhea to try and re-establish the balance of the microbiota, which is the complex community of microorganisms normally in the bowel, to hopefully re-establish health and fast-track management of the diarrhea. And in terms of diarrhea, I'm sure we're all familiar with it as being a common problem in foals, and up to 60 to 80% of foals, depending on the year, less than 6 months of age, will be affected by diarrhea. Now, as the flow diagram on the right, some of these are non-infectious and of less importance, but others are more problematic, particularly those with an infectious nature, either being bacterial, viral, or parasitic. And it's those infectious causes of diarrhea that can have real impact, both in the terms of the individual foal. And here's an example of a foal with marked clinical consequences of diarrhea. And that's at the individual animal level, but we've also got considerations around how diarrhea may spread between animals, and so we can have outbreaks on the farm. But we also need to think about ourselves, because some of the agents of diarrhea in foals are what we call zoonotic. So humans can directly or indirectly also be infected with those pathogens. And a good example would be salmonella, and we certainly have endemic issues with salmonella in Australia in our horse population. And when we're thinking about infectious causes of diarrhea, what we start to get concerned about are disruptions to the normal balance of the bugs in the bowel. And so we're talking about the microbiota, and the fancy term that we use in terms of that disruption is dysbiosis. And when we have an animal with, and this is for a foal or an adult, truth be told, when we have an animal with diarrhea, particularly a foal, we're thinking about what are our treatment targets? How can we manage this condition? And so we often use anti-inflammatory treatments. These animals often have fluid deficits, and so we have targeted strategies in terms of providing intravenous or oral fluids and nutritional support of the animal. We like to think that intestinal protectants may be of benefit, where we can almost form a barrier on the lining of the intestine to lessen the likelihood of damage, but that may be wishful thinking. And we often do resort to the use of antimicrobial drugs, but when we think about that, that doesn't necessarily allow management of that dysbiosis. And so here's a schematic of lots of bugs in the bowel. And so we need to think about how we can best address that and try not to overuse antimicrobials, so antibiotic drugs, because that's going to increase the risk of certain complications, either clinical, but also increasing the risk of the prevalence of antimicrobial associated resistance. So what do I mean by the fecal microbiota? Well, as I said at the start, this is the complex population of microorganisms, bacteria, viruses, protozoa, etc., that normally are located within the alimentary tract. It's the most genetically diverse population on the planet, and we're all carrying them around inside us. So we've got this rich and diverse population, as schematically demonstrated up in the top right there, and this microbiota has a really important role, and it's been investigated extensively in the human healthcare settings, where the microbiota is so important for intestinal health, but also health beyond the confines of the alimentary tract, immunity and brain health and other organ and endocrine function. So it occupies a really important role. In terms of the setting of this project, what we're really focusing on is the roles in terms of regulating gut inflammation and the risk of diarrhea. So we have done some work. There is work ongoing in looking at the microbiota in horses. And as you can see, there's a number of studies that have been published, and we're continuing to get new studies. And these studies are providing really useful foundation information. We're getting an understanding of what the population looks like. You know, it's not just one individual bacteria down there. We're dealing with a very diverse population. And so if we look at that schematically, instead of thinking about individual microorganisms, Salmonella, E. coli, whatever it may be, we need to start thinking about them as this big interrelated community and population of bugs in the intestine. We're now also starting to get an appreciation of how disease can disrupt that. And again, we're riding on the coattails of human medicine, where it's clear that there's a whole raft of conditions that can disrupt this normal microbiota. And one of those in the horse that we're starting to get evidence of is diarrhea. So both foals and adult horses with diarrhea, we know will have a disruption to that microbiota. But we don't have a good understanding of the effects of treatment, either inadvertent or deliberate attempts to alter that microbiota. And if we think about antimicrobials, we use them, and they are an important part of our arsenal for a whole variety of treatment strategies in any healthcare setting, including horses. When we use this to target a very specific microorganism, let's say Salmonella, for example, we've got Salmonella in the crosshairs, we're using a drug that we know based on laboratory testing should get Salmonella. And when we look at the microbiota in the alimentary tract, we might take out Salmonella to a degree, but we may also take out non-target bacteria as well. And so that can actually further disrupt that normal balance of the bacteria in the bowel. So it could actually accentuate that dysbiosis. Conversely, what we're now moving towards is, well, can we try and re-establish the balance of that very complex population of bacteria in the horse's bowel by transplanting manure from a healthy horse that's hopefully got the bacteria in all the right balance to address that disruption that the underlying condition has caused. We've got some evidence based from recent studies published in the literature that in adult horses with diarrhea, admittedly small numbers, but in adult horses it seems to have a beneficial effect in terms of improving the microbiota and improving clinical outcomes. So our objectives in this study was to look at fecal microbiota transplants, and so the easiest way to think about it is a poo shake, and look at that in three settings. What's the safety of these treatments? What are the clinical effects? You know, are we getting a beneficial outcome in terms of diarrhea and clinical resolution? But also, what's specifically happening at the level of the microbiota? Are we changing that population in our favour? We also want to get some idea of what the pathogens that are causing diarrhea in our foal populations in Australia. So we're sort of getting some initial data on, okay, who are the main culprits that we're having to deal with? And then with these treatments, we need to be mindful there potentially could be complications. So are we seeing any complications? And the microbiota transplants, that's not a new concept. And whilst it's not very sexy science, so to speak, it's actually a very useful treatment for certain conditions in human medicine, and in particular Clostridium difficile. It's a problematic bacteria that can cause long-standing and very problematic inflammatory bowel disease syndromes in humans. And fecal microbiota transplantation has been shown to result in really good outcomes in those settings, and indeed in other clinical settings as well, in human gastroenterology. So there certainly runs on the board from a human healthcare perspective. Now we need to see whether we're going to see that in horses. So how do we go about it? We've been recruiting foals with diarrhea and then randomly assigning them to either control, so they just get saline as a specific enteral treatment, or they're getting the so-called poo shake, where we collect feces from a healthy donor horse, we we blend it up, we basically get the liquid, and then we administer that by nasogastric tube to the horse, and we do that to the foal. So we do that 3 times, And at each time, we also collect a fecal sample to then do some more sophisticated laboratory work, which we're about to embark on. So that's where we actually extract the DNA, the bacterial DNA from the sample, and using laboratory methods, we can actually sequence the genes. And so we can actually get a very sophisticated analysis of which bacteria are there, what proportions, and also how they change with disease, and also how they change with treatment. And so sort of looking at both the effects of the disease, but also the treatment effects on the microbiota. So the results to date, we've got close to 26 foals that have been recruited, and from those foals, we've identified a number of pathogens, and I've just listed them on the slide there. Cryptosporidium, which is a parasite, certain bacteria, including Salmonella and Clostridial species, and also rotavirus. And some of these foals have actually had infection with more than one pathogen. So we've had, for example, foals that have had both Cryptosporidium and rotavirus. And so there's a concern that may have an amplifying effect on disruption to the microbiota. We've had no adverse effects in foals that have been treated with a microbiota, and that reflects also our experiences in adult horses. So it does seem to be a safe treatment. If we think about it, foals, as part of their normal development, will actually eat mares' feces, and so coprophagy is the term, and that's really establishing the normal flora and the bacteria in their intestines. So it's encouraging that we're not seeing any adverse effects. From a clinical effects, the interim results, we haven't done all the data analysis yet, but certainly in some animals that have been treated, we certainly get the sense that there's an overall shorter duration of the diarrhea. So it may facilitate resolution. I need to hasten to add, these foals are also getting managed according to their clinical need, with intravenous fluids, anti-inflammatories, treatments for toxemia, etc. So they're not just getting the poo shake, they're also getting the other standard components to their management. And we're also seeing improvement in clinical signs in some of the laboratory data. And so that's cause for cautious optimism, but we need to be mindful we haven't analysed all the clinical data. We've still got some further work to do to get greater sort of scientific assessment of the effects of both the disease but also treatment on outcomes. This month we're about to start our sophisticated laboratory methods as far as the DNA extraction and PCR analyses, and getting in a position where we're going to be able to do our gene sequencing and progress through to the more sophisticated data analysis. So that is still pending. In terms of the practical application of these results, and I appreciate this is a real whistle-stop tour for this project and what we've found so far, but I think fecal microbiota transplants certainly should be considered in the management of foal diarrhea. A few things that are important around that, though. It is important to identify a good donor horse, and so we've done that in our colleagues that we're working with and other practices, where we've got a resident horse that isn't being treated with antibiotics, doesn't have any clinical abnormalities, and are also screened for important pathogens that could be transmitted through feces, so certain bacteria and viruses, etc. So that's important to be able to do that. And we do that every year. We screen our donor horses, and this may also lead to an opportunity for us as an industry to reduce our antimicrobial use in diarrhea cases. So what's the overall benefit and impact that we anticipate is going to come out of this study? We're certainly expanding our understanding of the microbiota and also dysbiosis in horses, and we're using very established and sophisticated methods. And this is going to hopefully lead to evidence-based advances in our management of foals with diarrhea, and that's of relevance to all stakeholders within our industry, our breeders, our veterinarians, our funding colleagues, and also the more broadly in the community at large in terms of assurance of improvements in animal wellbeing and health. And if we can move to reduce antimicrobial use, that's hopefully also going to lessen the selection pressure for antimicrobial resistance, which is a big problem both in veterinary medicine but also in human medicine. So just to finish off, I also want to draw acknowledgement to the staff and students at both the Veterinary Clinical Centre where I work at Sturt University, and also the diagnostic laboratory here who's done some of the screening work, and also colleagues who are contributors to the study in case recruitment, both at Scone Equine Hospital and also South Eastern Equine Hospital. So I'm going to stop sharing now, and I'm I'm very happy to entertain any questions.
Excellent, thanks, Kris.
Yes.
Jenny, you have a question.
Yes, thanks. Very interesting. Two things. You said 20% of the infections are infectious. I'm just interested what loss of life or what the impact of that bad infectious one is. And two, I'm interested if any other species of animal. I know in livestock and cattle particularly, salmonella can be a major problem, and I don't know if any of the other species have done similar work and what the outcomes have been. Thank you.
Yeah, good questions, Jenny. And in terms of those figures that are provided are pretty rubbery, and look, they can vary across populations. In terms of the more problematic causes of diarrhea, in salmonella, for example, some of the clostridial causes, look, that can be fatal, absolutely, and we do lose foals to those conditions. Not every foal with salmonella, not every foal with Clostridium difficile, for example, is going to succumb to the condition and die, but they are more challenging in terms of therapeutic management and the intensive requirements for treatment, and that also then extends to greater costs. Look, in terms of mortality rates, I would say dependent on the condition in the year, we might lose 10%. Your other question in terms of other species, that's a really important consideration, and it is done in ruminants. And again, ruminal transplants are done in cattle and for a very similar reason, to try and re-establish the normal balance of the microbiota within the rumen. So the rumen in a ruminant, in particular cattle, but also sheep, this has been done, the transplant, that's their main fermentation vat. Whereas in a horse, it's more the large intestine, so they're actually at the other end of the alimentary tract. But it certainly has been established as an effective avenue to manage certain types of disruption to the bowel in cattle. There's a little bit of work in other species as well, including dogs. So there's some evidence in small animals that this may be an avenue in which we can modify to our benefit what's happening in the alimentary tract.
So welcome everybody. This is just an update for anyone who saw the webinars last year. Then we sort of introduced our research project and introduced all our team members, which is a team of pathologists as well as a team of epidemiologists that are doing this project on basically fetal postmortems, but trying to improve things. So the major objective for this project is to reduce pregnancy loss, reduce abortions. So we get more of, I'll just get my pointer up, more of these are the guys, and a whole lot less of these guys, because that's not much fun. So we do this at two basic levels. We want to look at the individual mare level, and by improving the understanding of risk factors on the farm and for the various different causes of pregnancy loss, we can start doing management changes that will prevent further losses. But the other way is also at an industry level to detect outbreaks quicker and get in and set up systems to reduce the losses of that ongoing outbreak. And to do this, we need a really good surveillance system. So those are the two primary techniques that we're using to work on this system. So we'll be looking at, when we do this, we need to look at the various areas in that chain of events that occurs when we get a pregnancy loss. First the post-mortem, then the lab testing, and the data analysis and the feedback to the farms to let you know what's actually going on. So in this area, we're working on training vets. We have developed some really good reference tools for the vets so that they can help make the diagnosis, as well as in the laboratory area. We've also, once again, we're going to be training pathologists, and we've developed some really good reference material as well. And then the epidemiologists are mostly working in the data analysis, and we'll go through and discuss all those in just a little bit more detail. So first of all, what's the team of epidemiologists doing, and myself, who's working mostly with them? The first thing we did was we've created visual atlases for the vets and the specialist pathologists, and this is to provide training material, to provide material for reference. And also we're developing a training program for veterinarians so that vets are upskilled in doing fetal postmortems. And I am working really hard on creating a really good database system for recording all the information that we collect. So in these first ones, we've created this visual atlas. First thing we needed to do was list out all the causes there are of equine pregnancy loss in Australia, so that we cover all the things that the vets might need to be looking for. And then we needed to go through really carefully, the team of the three pathologists and myself, go through really carefully and describe exactly what each of those causes of pregnancy loss are going to look like, and create tips for the vets, so that a good summary for them so this is what a herpes is going to look like, as well as lots of tables so that they can have quick references. The other aspect to it was to make sure we've got lots and lots of pictures. Once again, the best way when they get for the vets to get a good idea of what they're going to be looking for when they're doing the postmortem, lots and lots of good pictures is very, very helpful. Then we went through and did the same thing for the pathologists. So basically a really nice textbook for pathologists with really close detailed descriptions, as well as lots and lots of images, lots of good tables, lots of tips for them, so that they've got a good idea of what they're going to be looking for when they get the samples to them from the vets. So those are virtually, the first draft of the first edition of them is completed. As we go through in the finishing off phase 1 and moving into phase 2 of this project, we will be refining those based on the feedback we get from the vets and from the pathologists. So they're 99% complete, but they'll be tweaked to improve them even more for the vets and pathologists that are going to be using it. Another aspect of the work that I'm doing is working out a system to train vets. And to start with, I've started with the Scone Equine Hospital vets, and during 2020 and then 2021 particularly, I've been training the vets at Scone Equine Hospital to do fetal postmortems. And actually they've been absolutely wonderful guinea pigs for me, in that they've been providing feedback about how that training's working, and so that we can develop an efficient system that can be rolled out across the country. Another part of the work I'm doing is to get a secure electronic reporting system. We've got a system that is basically an app that goes on an iPad that the vets can use while they're doing the postmortem, and it's a lovely system in that it helps guide the vets through each part of the postmortem, collecting the history, looking at the outside of the foal and abdomen. So they've got advice and help to sort of go through, know what to look for, and then record the various different things that are important, as well as an opportunity to take lots and lots of photos, which is really, really helpful if they're wanting to look for any assistance in making the diagnosis. I can have a look at what they've collected and give them much better advice. But the other aspect of this system, if we're going to use it to be recording fetal postmortems from multiple places in Australia, we've got to make it really secure. It's absolutely crucial that it is only the vet doing the postmortem and the farm that submitted the fetus have any knowledge of the mare's name and the farm. And so what we're doing, I'm working with an app developer as well at the moment, and we're working out a system where for the one that's not yours will be coded. They all get a code, and you can only see the cases that the vet can only see the cases that they're actually working on. So that's getting close to being up and running, and we're hoping to use it in the phase 2 of the project that we'll be talking about a bit further later. So the other part of the team are the epidemiologists, and they're working really hard at developing the baseline data that we'll need to start with for the surveillance system. In order for this surveillance system that we want, so the surveillance system is so that we can identify when we're getting an outbreak of abortions and what's causing it, so we can get on and start trying to control the problem. But for that to work well and automatically, which is what we need it to be an automatic system, you need to have good baseline data of knowing when and where abortions normally occur, so that when there's a blip, the system automatically notifies the people that are responsible for initiating basically an emergency response. So we've started with the ones in the Hunter Valley, and they've created these baseline graphs to see what happens. So when during the year, how many, and at what stage of pregnancy these losses are occurring. So they've got that started now, but that's going to be rolled out further. So in the chain of events of the pregnancy loss investigation, this is the area that the epidemiologists work predominantly. I do a little bit of this work in data analysis in developing the database itself, but doing the analysis and getting all the baseline stuff so that we can actually do an effective and efficient surveillance system, that's the job of the epidemiologists. So they've already done that baseline spotting. Now we've got to refine it and develop the system. And in order to do that, they're going to need to do 2 projects. The first project will be to find out a little bit better what actually happened last year. Really, it's going to be the best way to do it. So they will be wanting to find out how many losses occurred, when they occurred, and what stage of pregnancy they occurred. So what they're going to want to do now is, what they want to do is send out a questionnaire. So we'll be looking at farms throughout the east coast of Australia and asking you what happened last year. Now this will be collected in a completely anonymous way, so nobody will know exactly which farm or at what time abortions occurred, but we'll get this overall normal pattern, so we'll know what happened last year. And that's sort of the beginning of getting the information. So we've got a baseline to start working on what is basically an artificial intelligence system. And that's where we're looking for participation from the start. The data that we get and how good the surveillance system that we can set up will be dependent on how good the data we get back from you. So when you're invited to participate in this questionnaire, it will be, you are contributing and will be contributing enormously to improving and reducing the number of losses due to abortions. The other study that will be happening, which is going to work closely with phase 1 and phase 2 of the project, is what we call a case control study. Now, a case control study is where you get a case, so an abortion, which will be a case, and then you look at 2 other mares on the same farm that didn't abort, and you'll ask specific questions about the case and get the same information about the controls, the mares that didn't abort, and both sort of factors like mare factors, what the cause of the abortion will be recorded, and environment, where she was living, what she was eating, what time of year, what the weather was like, a whole lot of details like that. And you'll compare between the cases that did occur, the cases and the controls, and see what was different. And this will give us what we call risk factors. And once you know risk factors, you can go about reducing them. And if you reduce your risk factors, you reduce your abortions. So it'll be important for the overall surveillance system, but it'll also give you information that you'll be able to use basically next year, so or the year after. So you'll have information that will help you start to reduce your pregnancy losses pretty much straight away, even without going through and developing a surveillance system. We have completed, the epidemiologist led also one part of the project in the early years of this, and which was the scoping review of the literature, which is, we went back for 60 years of the literature on pregnancy loss around the whole world to see what was reported, what was common, what people were most concerned about, and what patterns were changing. And this paper's just been accepted, which is a tremendous achievement. And what we found were that of the infectious causes of abortion, herpes was the most commonly reported. So that was the one that gets researchers most excited and most concerned. And certainly back in the 60s and 70s, there was a great deal we needed to learn about herpes. And as a result of it, the incidence of herpes has reduced quite a lot, particularly in the major thoroughbred breeding areas of the world with vaccination and really good biosecurity. The other thing that was interesting about that is that leptospirosis, which is a zoonotic disease, is quite a common cause of abortion around the world, but it isn't one we see in Australia. So that was quite interesting. And so, but I do think we need to keep an eye at that. That's something we need to keep monitoring, and would be one of the really useful tools of having a surveillance system, is that we would be able to pick up if we suddenly start to get leptospirosis. And that is really important, because leptospirosis is a dreadful disease for people to get, as well as obviously being a disaster for the pregnancy. Interestingly, a couple of other zoonoses that have cropped up here in Australia, the one Coxiella, which is Q fever, and Chlamydia psittaci, which we've had some outbreaks of, are also being increasingly reported throughout the world, which is quite important, because once again, the people, when you're dealing with an abortion, you're frequently associated with a lot of infected fluid, and it's very hard to keep things under control. So we need to understand what risks there are to people, because both Q fever and psittacosis can be really, really serious diseases. And we certainly don't want to see people getting very sick with that. The other thing that we discovered is that, as for everywhere else in the world, we're similar to everyone else in that umbilical cord torsion is the most common cause of non-infectious abortion. It causes probably 20 to 30% of all pregnancy losses, and it's similar, and that's similar to what's reported in the rest of the world. So what's our next step? Basically, it's helping with completing our phase 1, and that will be supporting the epidemiologists in their questionnaires and their surveys. That will be really getting that information is what we are wanting to get for your participation, because that will give us the best opportunity to get great information, and that will then allow us to provide you with the best advice that you can get in order to reduce abortions. And I just want to remind you that this data will be completely anonymous. No one will know that it was your farm that had any abortions or which mares it will be. But we will be able to sort of determine what region, so it'll be done regionally rather than, you know, by farm, or definitely not mare by mare, so it'll be just completely anonymous if you participate. So I'd really encourage you all to participate. But the really exciting bit is the step on to phase 2. So the groundwork is, phase 1 is basically the groundwork, getting things set up, getting, you know, the abortion system set up, starting to train vets, getting the electronic recording system set up, and then getting that reference material, it's all established. So that is, you know, really has progressed well, and we've got the vast majority of that done. It just needs to be refined. And we were, you know, planning on refining it this 12 months. But with the opportunity of phase 2, we'll be able to really nail down where the problems are and get a system that is working really well. So it's the wonderful opportunity that we have got funding now that will be able to support the investigation of up to, I think, 250 fetal postmortems over the next 2 years. So this means the abortion investigations will be done at no cost to the owner. That's the really wonderful thing. And but we will use it to go into this research system. So the vets, so what we'll be doing is training specific vets in Queensland, New South Wales and Victoria. So we'll continue training the Skynet One hospital vets, but we'll we've got a practice in Victoria, a partner practice in Victoria and a partner practice in Queensland, who we are training them up at the moment to be able to do fetal postmortems. And the other thing we'll be doing is training the pathologists in the labs that the work goes to, so that they can recognise what the major causes of equine pregnancy loss are, and can give the vets a really good idea of, you know, and give them some advice and help. And the aim of this is so that we can find out if there are any problems, and particularly one of the things is we've limited it, unfortunately. If you want to participate in this system, the samples have to go to one of these 3 practices. And we're doing that so that we keep control of it, because at the moment we've got to test everything, and particularly test the security of the system. And so we don't want information that, you know, private information leaking out. And we think we've got the system working, but just in case there's a hit, we want this in a very controlled system, so that we've got complete confidence that when it's ready to roll out, it'll be completely secure, and the system will work well, and it will be able to roll out more easily. So with phase 2 is this initial area of Queensland, Victoria, one practice in Queensland, Victoria, and New South Wales. The ultimate aim is to get it to go Australia-wide. And so that's the purpose in the next 2 years in phase 2. Now, the TBA have come on board to help support this and help administer this. And James is going to talk a great deal more about what their role is and give you a lot more information about phase 2 and how to participate in it. But that's my end of the phase 2, that I'll be doing the work and trying to train the vets and get the system working really well. And James will tell you how they're going to get their system working really as efficiently as possible. So that's all I need to participate in, and I'll need to stop sharing.
Thank you, Joan. That was a great insight. Do we have any questions for Joan? I'm sure there'll be a few. If we can get someone to raise their hand or pop a question in the chat box. Carl, did you have your hand up?
My wife did, Justine.
Oh, you're okay. Hey, Justine, how are you tonight?
Good, thank you. Well, just a couple of quick questions, Joan, if I could ask them. And that was a very informative session, and I wish I'd seen last year's presentation as well. With regards to the risk factors, are you including stresses that may include particular weather events or weather conditions as some of those risk factors? And then also, is it only applicable to thoroughbred breeds, or is this something to extend right across any stud who's breeding any type of horse in terms of infectious herpes and those sorts of things? And then At what stage in the pregnancy are you considering it an abortion, and is it only if you find the fetus, or is it if you know, you get to a certain point past beyond into the pregnancy, and you just describe it then as a reabsorption instead of an abortion?
Great question, and it covers a lot of the various different parts. Some of it's the epidemiology. Now, you'll have to remind me of you first. I'll answer your last question first because I can remember it. We might have to go back and check. But we will be, the epidemiologists will be looking at when the abortion occurred. So that questionnaire will be finding out whether the abortion was seen, whether there was a diagnosis, and how many pregnancy losses occurred without it being seen. So those mares that have say a 45-day pregnancy, positive pregnancy test, and then when you preg test them in May, June, July, they come up empty, but nobody saw anything in between. They'll be included, and they're not part of phase 2, because phase 2 is the investigation of all the abortions. But the epidemiologists in that questionnaire will cover those sort of topics. The first question, I think, was weather. Is that right, Justine? The weather.
Factors.
The risk factors will be as wide as we can make them. It was interesting. We've just been working on developing the questionnaire, and one of the things that really worried me about it was how long it was, because there were so many questions we wanted to ask. So and a lot of them are the weather. The other thing about it, as we get at what time of year they did occur, each abortion occurred. And we did this with the Hunter Valley abortions that, that the epidemiologist says, what time of year it did. You can get pretty good data from the Weather Bureau, from the weather stations, of what the prevailing weather was as well. So yeah, that sort of factors in with the epidemiologist. So we're definitely looking at weather forecasting, weather events, and whether it had been a rainy year, a wet year, a particularly dry year. So yeah, those are really important factors. Now there was one other question, and have I answered all your questions? I think
it was just whether or not it was extending to all horse breeders or simply narrow to the thoroughbred breeding at the moment.
Well, at the moment it's supported by the Thoroughbred Breeders Levy. So this is restricted to thoroughbreds only, because it's being paid for basically by thoroughbreds. And the AgriFutures is a thoroughbred research project. So right now it's restricted in that way. So the funding is restricted, but the information that we've got, those resources on how to do a post-mortem, what to look for the vets that are doing them, that will be available that'll be freely available once it'll go up on the AgriFutures website, and all that information will be available. And the other information is the results of those questionnaires and all the details that we find out about when, where, how, what risk factors they are. Once again, that will be available for all breeders in Australia, but the actual abortion investigations in the next 2 years, phase 2, that is limited to thoroughbred mares.
So there's nothing similar to this particular project that's being conducted internationally, for example, through the Irish Stud?
Not that I'm aware of nationally. There are different systems in the UK and in Kentucky, particularly. Kentucky are really lucky in that they have a sort of surveillance system, and that all their abortions are funded by the government, and they go through their veterinary diagnostic laboratory. So that's, they do all breeds, and it's always free. So that's just an incredibly privileged existence for them. And they have a bit of a surveillance system, but at the moment it is just restricted to Australian thoroughbreds. And if the other breeds are keen to do this sort of thing and participate, then it'll be just a matter of coming in and discussing things and trying to bring the funding in is really what, it's whoever pays the piper gets the work, really. And the thoroughbreds have got themselves sorted and organised for it. So it'd be great to see. I'd love to see the other breeds doing it.
The dairy industry is pretty sophisticated and quite advanced in terms of their breeding program. They actually run a computer-generated model that hangs around their cattle necks, and they can tell when the cow's in heat, what its production is, a whole range of other things if it's got a temperature. Is that likely, the surveillance program that you're talking about, is that something that could actually extend that far, that rather than as simply having a collar with a tag on it around our broodmare's neck to be able to differentiate them in a herd, that you could actually take this sophisticated system and engage something that ends up being microchipped related to a horse and they wear it?
I think that would be a brilliant system to get established, because it's certainly in both the beef cattle and the dairy cattle industries, it's really starting to, those sort of surveillances are really starting to take off. But at the moment, we're just, looking at picking up the abortion basically and starting off. But yeah, it's got enormous potential. Once you've got a surveillance system built, there's enormous potential to build on it, because you've got the framework. And with the support of AgriFutures, we could continue doing further research to sort of build other and add other automatic, because we'll have, certainly it's there. And once you've got the framework, it's much easier to add on to it. So, yeah, I think that'd be a great idea. Do we have time, Annalise, for a question from Catherine? Looks like she's got a question.
Oh, yeah, all right, this once.
Just for you, Catherine.
Why, thank you, Joan. Thanks, Annalise. I really just wanted to make sure that everybody on the webinar understands what an extraordinary resource has been gathered by this project. And to my knowledge, there isn't anything like this that's been got together, all of the information in the atlases across the world. So it will be a resource which actually serves the thoroughbred industry and the horse breeding industry across the world. And I think that's something that should really be applauded. It's been a phenomenal effort by the team, and I'd just like to congratulate you on your good work, Joan. Thank you very much. Thank
you. It's certainly been a team effort, and what's been the best part of it is just having this wonderful team that works so well together. It's energising and enthusiastic, and I've learned so much. It's great. I've really loved it. So yeah, thank you.
I will now welcome James Murray of Thoroughbred Breeders Australia. How are you, James?
Good, thanks, Charmaine. I just thought to begin with, I just again echo the sentiments of Catherine there. Just a fantastic presentation by Joan, and a lot of hard work and long hours go into that. But hopefully, it's built the foundation to really build into the future.
Definitely. Now, James, you might be a new face to some in the industry. You're the marketing and projects coordinator for TBA. How long have you been in this role for?
Yeah, so I've obviously joined Tom here and Cecilia at TBA in the last 3 months. Coming off, I guess, a horse sort of background, was in the Hunter Valley for the last couple of years working on a farm. But yeah, really great to be a part of the team, and I've met quite a few breeders already, and I'm sure, I'm looking forward to getting to meet a lot more of you, and hopefully tonight I can shed some light on TBA and our role in phase 2 and explain phase 2 with a little bit more detail.
Well, James, thank you for that introduction. I might let you jump straight in and explain the part that you and TBA will play in this role. So I'll get you to kick off from there.
Yeah, fantastic. Just give me a sec. Hopefully everyone's got that set up. So just to begin with, phase 2, obviously Joan touched on it, is the post-mortem of late-term pregnancy loss. So just to build the foundations and start with, I just wanted to touch on how AgriFutures and Thoroughbred Breeders Australia have kind of come together. So obviously AgriFutures is a government-established research and development corporation. And that's really come from TBA when we've successfully lobbied the Australian government in the past to match the Thoroughbred Research and Development Levy. So for every mare bred in Australia, there's a $10 fee paid by the mare owner and the stallion owner. And then this is matched starting in 2017 by the federal government. So each year that equates to up to nearly $1 million per annum, which is a fantastic resource and a huge amount of money to go towards R&D each year, and ultimately is funding a program which I'm about to delve into a little bit more. So obviously with this Thoroughbred levy, there is an industry advisory panel with people such as Mike Becker in Victoria, Michael Grieve in Queensland, Derek Field for Whitten, and these are the type of people, included as well as us, we're kind of listening and making sure this money is going to the right places, which kind of leads me into this project, which is abortion. And obviously the massive loss, the feedback I should say we're getting is the massive loss that abortions have on the thoroughbred industry year in and year out. So Joan touched on phase one, how it's being successful in the questionnaire, and obviously that was just done in the Hunter Valley region in New South Wales. But with phase two, we've expanded that to southern Queensland and northern Victoria, so really expanding our reach. And the two clinics chosen outside New South Wales, which she also touched on, are Darling Down Vets, which is in Queensland, just outside Toowoomba, and then Goulburn Valley Equine Unit in Shepparton, Victoria. So I really want to touch on why this project is important for the industry and then you guys as breeders as a whole. If you take one thing out of tonight, I'd hope it to be this. So as I said, so much of the feedback we're getting is how costly abortions can be. And that's why these, on a micro sort of scale, these free postmortems are a massive plus to any farm. In particularly, I've been hearing especially farms who build their business on agistment and have agistment properties. If you've got a small amount of mares and a client doesn't really want to fund the post-mortem, it kind of is put back onto the farm or the business itself. This phase two and these funds from AgriFutures are really there to help these farms out. And then linking into that, hopefully by doing these post-mortems in a micro scale, you can catch those abortion storms that can happen on farms. From a larger perspective and looking at a macro sort of point of view, the greater good of the industry is here, which Joan kind of touched on, and hopefully by creating this database and in the future as we grow and build to a national scale after these pilot stages are complete, we can really gain a deeper understanding of the industry through those trends and build a greater understanding on the causes. And this, hopefully, fingers crossed, will then lead to a long-term decline in abortions in the industry, which is a success for us. Well, from our perspective, a success, and I'm sure as breeders you can agree that'll also be a success. I really want to emphasise and touch on the role of TBA in this second phase. So we are here to make this as easy as possible in simple terms for you guys as breeders. We are here to communicate, we're here to do the administration, but that is where it ends. We want to make it as simple as possible, so we'll do all the payments, we'll help facilitate understandings, which will start to push in the coming weeks, including information packs with more details, and help AgriFutures with administrative side. But we're simply here, in simple terms, I should say, to make it as easy as possible for the breeders and make this as smooth as possible. Joan really articulated the confidentiality. I just want to, again, point out that TBA will have zero access to any of the data and any of the regions. We're simply there just working out the invoicing and the payments and helping breeders involved with the project. From a TBA sort of point of view, I wanted to point out some of the clear objectives. So obviously we want to provide breeders with the funds to undertake these postmortems. If breeders have access to them, hopefully they'll get better management of their mares and then in the future obviously help their farms and their business as a whole. On a grand scale, this data collected to identify trends and causes in the late-term pregnancy loss is going to help and prevent those mid to late-term pregnancy losses in the future. And then finally, again, just recapping what Joan has said, just building that national surveillance system to really help out the industry as a whole. I know that was sort of brief, but hopefully that gave a greater kind of understanding. In the next couple of weeks, we're really going to make a push to push out those data packs with more information, how to get involved. If anyone has any questions, I'm here, and yeah, thank you so much for listening.
Thank you, James. That was great to hear about TBA's role in the project. Now, does anyone have any questions for James? Not currently, unless I can't see someone. Well, if you do, James obviously has his details that were up on screen, or you can jump on the TBA website and get in touch with him directly there. But I'm sure you're going to hear some more from James in the coming months as the project really gets underway.